We have previously discussed how glucose gets broken down into two equivalents of pyruvate during the process of glycolysis. In this video, we're going to discuss the fate of the pyruvate molecules when there's plenty of oxygen around for aerobic respiration. The pyruvate that gets produced during glycolysis first needs to enter into the mitochondria. This happens through a specialized type of protein that transports pyruvate into the matrix of the mitochondria. This protein is called the pyruvate carrier protein. Once inside the mitochondria, there's a really large enzyme complex known as pyruvate dehydrogenase, which catalyzes the oxidative decarboxylation of pyruvate. The resulting 2-carbon unit, which is essentially acetic acid, is conjugated onto coenzyme A. Okay, we call this acetyl-CoA, and this is the entry point into the really important TCA cycle, or tricarboxylic acid cycle. The first step of this cycle is combining the 2-carbon unit from acetyl-CoA with oxaloacetate using an enzyme known as citrate synthase. Okay, oxaloacetate is a 4-carbon unit, and we merge this 4-carbon unit together with a 2-carbon acetyl group from acetyl-CoA, and we end up with a 6-carbon tricarboxylic acid. Okay, this is known as citrate or citric acid. Okay, citrate then gets isomerized. We simply move the position of the alcohol group from C3 to C4, okay, and we go from citrate to isocitrate. Once we've produced isocitrate, we go through our second oxidative decarboxylation process. Okay, so in this step, we remove electrons from isocitrate, and those electrons are given to our important redox cofactor NAD+. So NAD+ gets reduced to NADH, and in the process, we lose carbon dioxide from isocitrate. This gives us a molecule known as alpha-ketoglutarate. Okay, and if you continue on in biochemistry, you will learn about the importance that alpha-ketoglutarate plays in an awful lot of different metabolic aspects of the cell. Okay, alpha-ketoglutarate then gets oxidatively decarboxylated to produce succinyl-CoA. So chemically, it's the same thing happening here. We are losing carbon dioxide, and we are reducing NAD+ to NADH. At this point, we have produced 2 equivalents of carbon dioxide. So we had 2 carbons coming in the form of acetyl-CoA, and we've now lost 2 carbons in the form of carbon dioxide. So this accounts for the total oxidation of those carbons. What we now need to do is to regenerate oxaloacetate from succinyl-CoA, and we have created a complete cycle. So the succinyl-CoA first undergoes a hydrolysis where we lose coenzyme A. So coenzyme A gets hydrolyzed off, and we produce succinate. This hydrolysis actually has, it's spontaneous enough that it can drive the synthesis of either GTP or ATP from precursor ADP or GDP. Okay, so in the image here, we just show the production of an NTP because it can either be ATP or GTP depending upon what tissue you're in. And the chemical energy that drives that is the hydrolysis of acetyl-CoA becoming succinate and CoA. Okay, once we have succinate, we undergo another oxidative process where we oxidize succinate to fumarate using an enzyme known as succinate dehydrogenase. Okay, so in this case, the electrons don't go to NAD+, instead they go to FAD, producing FADH2. Okay, this enzyme, succinate dehydrogenase, is a really interesting one because it's also an important enzyme in the mitochondrial electron transport chain. So in the next video, you're going to see this exact same enzyme, but we're going to call it complex 2. Its role is to oxidize succinate to produce fumarate, and in the process we generate FADH2, which then shuttles directly into the mitochondrial electron transport chain in complex 2. Okay, but now back to the TCA cycle. So we've produced fumarate. The next step is to hydrate fumarate to create malate, and then malate gets oxidized to oxaloacetate using malate dehydrogenase. This redox reaction produces another equivalent of NADH. Okay, we've now regenerated oxaloacetate, which can remerge with another equivalent of acetyl-CoA, and the process can start over. Okay, so now that we've looked at all of the important intermediates and briefly discussed some of the important enzymes, let's focus in on a couple of the other really critical aspects chemically that are important. Okay, one is the entry point into the TCA cycle is production of acetyl-CoA. The conversion of pyruvate to acetyl-CoA produces one equivalent of NADH. So before we even get into the TCA cycle, we have produced one reduced cofactor. Okay? During the TCA cycle, we produce another 3 equivalents of NADH. In addition, we produce one equivalent of FADH2. Okay, so if we put all the pieces together here, for each pyruvate, we produce 4 equivalents of NADH and 1 equivalent of FADH2. Okay, so remember that during glycolysis, we produce 2 NADHs per glucose. In addition, we produce 2 pyruvates. Okay, so let's total up let's figure out the total number of NADH and FADH2 that we produce per glucose. Okay, so we start with glucose, and during glycolysis, we generate 2 NADH. We also generate 2 pyruvates, which each of those 2 pyruvates get converted to acetyl-CoA, giving us another 2 equivalents of NADH. And then the acetyl-CoAs each enter into the TCA cycle. So for 1 glucose, we get 2 full rounds of the TCA cycle. And during each round of the TCA cycle, we make 3 NADHs for a total of 6 NADH, and we make 1 FADH2 per cycle for a total of 2 FADH2. So this gives us a total of 10 NADH yielded per glucose and 2 FADH2 yielded per glucose. Each of those reduced cofactors will now feed the mitochondrial electron transport chain to ultimately generate ATP.